My name is Bjarte, and although I work at a e-health company this week, I have stayed with the Department of Chemistry for 16 years. Prior to that, with the last 4 years with the Hydro Center for Quantum Molecular Sciences, which is exactly how scary as it sounds. I want to talk to you today about cold-adapted enzymes for science and profit. Now, the profit part was actually a lie. Sorry. I didn't profit from enzymes. but there is a biotech company in Tromsø that does profit from cold-adapted enzymes, so it's a white lie. So, enzymes are biological machines. They are super important. It's quite common to hear people talk about DNA being the recipe for life. And if DNA is the recipe for life, well then enzymes are life. Because enzymes are encoded by DNA. And anything that happens inside of you is a result of enzymes action. So many of you are drinking beer, consuming alcohol, and this is being detoxified in your liver by the enzyme alcohol dehydrogenase. Right? And perhaps there will be some snacks later, and you will have to start hydrolyzing some protein, some fats, and sugars. This will also be enzymes. So enzymes are super important. And we are all still fully dressed. Okay? Not that much beer yet. And that's nice, right? It's because we like to be warm. Humans are adapted to be at around 37 degrees internally, and we need to keep at that temperature. Otherwise, we get sick, right? Our bodies cease to function as they should because the enzymes are now out of their optimal temperature range. Now, if we were to go down to the harbor, which you shouldn't do after too many beers, and look down into the water, you might see fishes, right? And we know that the ocean is ice cold all year around here. But still the fishes thrive, right? And inside of the fish, there's still life, and this is still enzymes working. If you compare the enzyme sequences between me as a human and a fish, they are quite similar. And how can that be that enzymes that are so similar work at so different habitats? That's the question that I have been interested in answering the last years, and I will try to go into that. So, while we often use this Pac-Man figure I had on the previous slide to illustrate for beginning students how enzymes work, a proper enzyme looks more like this. So it's a blobby thingy that looks kind of weird, with all these crooks and creeks. And it's these crooks and creeks that makes enzymes work. For an enzyme to catalyze a chemical reaction, it needs to bind a small molecule, usually, into one of these crooks and crevices and kind of bend this molecule in such a way that it's more likely to undergo the chemical reaction that we're interested in. So how can we get these structures, right? It's not just a drawing. It's actually how it looks like, magnified some hundreds of millions of times. Enzymes are molecules. So that means that they are tractable by chemists like me that know chemical methods, because they are single molecules, somewhat complicated, but though simple enough. So we can, with some well-directed prayers, luck, and exhaustive testing, find conditions that encourage the enzymes to form these beautiful, beautiful crystals. This is a microscopy image that I took of one of my favorite enzymes, and you will be stunned, of course, by the beauty. But you will notice, once this fades, that there's symmetry to this as well. I won't name the symmetries. I could have at some point. But there are symmetries within this, because the enzymes are built up of billions and billions of single enzyme molecules stacked in a repeating unit, forming a crystal, not completely unlike the salt crystals that you would sprinkle on your food. So you really shouldn't do that. That would really annoy the ones that have made these crystals. People tend to spend a PhD trying to grow these. So once you have these crystals, what are you going to do about them? Well, the amazing thing about X-rays is that they can interact with chemical compounds. So the wavelength of an X-ray photon is so that it will bend as it passes through a crystal. And if you go to the medical office, the X-ray machine they have there is way too weak. So you want to go to a place where they have a proper X-ray machine. So this one is in France, and it's a kilometer long, or in circle. And how they work is that they take some electrons and they spin them around faster and faster and faster with these huge magnets, right? Towards the speed of light. Super fast, right? And then, once you have them spinning super, super fast, you make them turn into a beamline, as we say, and then you force them to stop very quickly. So they have these super excited electrons that are forced to stop, and they give off their energy as X-ray photons, and these are super, super energy rich, and you can focus them into this super narrow beam, like a 30 micrometer sized beam, the width of a human hair. So focused beam, and you can have it go through your crystal, and the X-ray beam will diverge so slightly going through your crystal, and this pattern that is formed as you rotate the crystal is enough to deduce the structure of the enzyme. So once you have that structure, this blobby thing that I showed you previously, what can you do about it? Well, here is a horrific answer, right? This is the Schrödinger equation. And I mean, it's just 4 letters, but it's oh so terrifying. So this is a Hamiltonian operator. This is the wave function. Now, I wanted to scare you, right? Just, I think that's fun, a good pedagogical trick. But the point of this Schrödinger equation, to me at least, is that it means that if we can describe the waves of the subatomic particles of the system, then we can also calculate whatever else that we could care for, especially the energy. And the energy is important. And a supercomputer makes that easier. So behold, there's an animation. So this is another one of my favorite enzymes, and it does this stupidly simple chemical reaction. It's beautiful to behold. It just transfers single hydrogen atom. This one, here it is. And once you let it evolve, it kind of jumps over here. Excitement! And what is beautiful about the quantum mechanics here is that since you know this and how the electrons usually distribute around this nuclei, you can calculate the energy of every one of these steps that occur. And I can go into the lab and I can measure properties of the system that corresponds to the energies that I can calculate. So that gives me a way to validate my system and calculate useful properties. Now, this is obviously just a toy system, so I'm going to scale it up because I was saying enzymes are big molecules with thousands of atoms, like this. And to add to the complexity, enzymes never exist in a vacuum. That would be weird. They do exist in water usually. That's a simplification that we make. They usually occur in cells. Those are kind of tricky. But you can have enzymes in water. That is a reasonable thing. And then you can introduce water molecules into your calculations. And since there's only so many supercomputers that I can use at the same time, we usually try to simplify our calculations in such a way that you do quantum mechanics in the center, where the interesting stuff is happening, and then you do regular Newtonian, like in Isaac Newton, dynamics for the rest of the system. So that speeds everything very much up. So by now, I hope that I have convinced you that 1, enzymes are alive, super important. 2, enzyme structure is the key to understanding enzymes. And that we can calculate useful things if we have a structure. How does that relate to the title of my talk? Not very clearly. So I'm hoping to take this now together for this final slide here. And that is that I have established that the technology that we have today is capable of answering a whole deal of questions. And we just need to find the way forward. And what I have on this slide here is my research proposal for the Norwegian Research Council for how to proceed. Because the chemical space of enzymes is immense. A simple enzyme has 100 residues, and there's 20 amino acids to choose for every one of these positions. And if you do some simple combinatorics, that's 20 to the power of 100. That's a big number. It actually surpasses the number of atoms in the known universe. Right? So you can't physically make a copy of every enzyme that you would be interested in. So we need to do smart samplings of this very large chemical space. And I'm proposing to do machine learning to deal with this. So I have built up a small data set of warm adapted, like me, organisms, and cold adapted counterparts, and some enzymes for those species. And I've tried to look at, kind of find a fingerprint of what is special for every class of enzymes. And the initial idea I had was just looking at what kind of pairs we see in the sequence. So we can see that these two sequences, they start with a histidine and then a asparagine. Well, it's the same in both of them, so that's kind of boring. The first one that is different here is a methionine in one of them and an isoleucine in the other one. Is that an important difference? I have no idea, right? But if we have thousands of different enzymes to compare pairwise on this, perhaps we can uncover a hidden signature. And I've made some averages here over the data set I have. And if I feed this into the computer, I can get quite high accuracies already in identifying a sequence as being either cold-adapted or warm-adapted. And I'm hoping to convince the Norwegian Research Council that this is the way to go, and that we can use the plethora of different other methods that I have to validate this approach. So, thank you very much. I have a QR code here. That is my latest publication. It came out last week. It's super nice. It's open access, so you can even take it right here. You don't have to go back to the university first. And feel free to ask questions. Thank you.